How to draw a nucleotide. That will be our focus of today's video after we go over some brief background. So first of all, nucleotides are the monomer building blocks that make up nucleic acids, the most famous of which is DNA. The slightly less famous is RNA. Now DNA is what we think of as a double helix. So it actually is made of two polynucleotide strands, or two nucleic acid strands, that are then hydrogen bonded together to hold the two strands together. And they are composed of varying arrangements of the monomer nucleotides A, T, C, and G. That's adenine, thymine, cytosine, and guanine. RNA, on the other hand, is a single-stranded polynucleotide or nucleic acid, and it is made of the nucleotide monomers A, U, C, and G. That's adenine, uracil, cytosine, and guanine. And they actually have a direct connection in that DNA stores the hereditary information for life and is used to pass that information from generation to generation of cells and generation to generation of organisms. But also within cells, the information on how to build proteins is stored within DNA, and they go between, for building proteins, is RNA, specifically messenger RNA. So DNA is used to transcribe and direct the cell on how to build RNA, so that's called transcription. And then ultimately, that RNA molecule is used to tell the cell how to build a protein, which will use just the amino acid peptide chain to illustrate. So that would be a protein. But there will be more on that later in the year. Now, one of the key components that students get confused about is determining what exactly is a nucleotide. They have a little bit of difficulty finding the nucleotide trees and the polynucleotide forest. So I've pulled up an image that shows a DNA polynucleotide chain, or nucleic acid, and an RNA polynucleotide chain, or a nucleic acid. Now, if we look here, we can see we've got thymine, guanine, cytosine, and adenine, uracil, guanine, cytosine, and adenine. Now, what I just drew an arrow at, though, is not the nucleotide. That is the nitrogenous base, which is one of three components when bonded together makes up a nucleotide. So now I'm going to go through and carefully circle each of the nucleotides of this polynucleotide chain, or the nucleic acid. So here's the thymine nitrogenous base, the 5-carbon sugar, which is actually deoxyribose in this case, and the phosphate group. So that is one nucleotide, and it's the thymine nucleotide. Again, the nitrogenous base guanine, the deoxyribose 5-carbon sugar, and the phosphate group. That is the second nucleotide. The nitrogenous base cytosine, the deoxyribose sugar, and the phosphate group, that's the third nucleotide in the DNA nucleic acid molecule. And then adenine, nitrogenous base, the 5-carbon sugar, deoxyribose, and the phosphate group. That is the fourth nucleotide in this DNA polynucleotide chain, or the DNA nucleic acid. Again, it is just a single strand. It is not the traditional double strand. And over on the RNA polynucleotide chain, let's go through this again. This is the uracil nitrogenous base, the 5-carbon sugar, but this time it's ribose, and the phosphate group. The guanine nitrogenous base, the 5-carbon sugar ribose, and the phosphate group. So 1, 2. Then the third one, this is cytosine nitrogenous base, the 5-carbon sugar, and the phosphate group. That's the third nucleotide. And finally, the adenine nitrogenous base, the 5-carbon sugar, and the phosphate group. That is the fourth nucleotide in the polynucleotide chain of RNA, or the RNA nucleic acid. So if we take a look here, we can see that a nucleotide is composed of 3 parts: a 5-carbon sugar, a phosphate group, and a nitrogenous base, which could be A, T, C, G, or U. These ones for DNA, and all but, and then A, C, G, and U for the RNA. Now, later on in the year when we're studying DNA replication and we're studying transcription and translation, we will look at these nucleotides again in much greater detail. But for today, we are going to focus on a basic drawing, which is a little more complicated than a very simplified drawing, but much less complicated than, say, what you would draw for an organic molecule class. So, I like to start with drawing the sugar because it's the center part of the molecule, and I like to draw the oxygen first. So, it's a pentose or 5-carbon sugar. So, there's carbon 1, there's carbon 2, there's carbon 3, there's carbon 4, carbon 5 goes up from there. So we actually write the carbon in this time, whereas the other carbons are inferred in the vertices of the pentagon with the oxygen at the top. And then it has 1 hydrogen here and 1 hydrogen here. So that is our 5-carbon sugar. It could be deoxyribose, or it could be ribose. It depends on what nucleotide we're drawing. And then we have a very simple way of drawing our nitrogenous base at this point. And that is just a simple little flag that you can write N base on, or you could write base, or you could write nitrogenous base. But at this point, you should write the full word, nitrogenous base, the full name. So the nitrogenous base binds on to the 5-carbon pentose sugar via carbon 1. And then the third component is the phosphate group. And the phosphate group binds on not at carbon 1, not at carbon 2, not at carbon 3, not at carbon 4, not at carbon 5, but at carbon 5. There's no more carbons left. So at carbon 5, the 4th bond holds on to the oxygen of a phosphate group, which then bonds onto the phosphate, which then we complete the rest of the phosphate group. So there's the negative charge, there's the negative charge, and then here is the oxygen double bonded to the phosphate. So that all together, those 3 components make up a nucleotide. And again, let's just highlight the different parts. So this right here is our 5-carbon pentose sugar, which could be deoxyribose or ribose. The phosphate group is right here. And the nitrogenous base is right here. So there are 3 subcomponents that make up a nucleotide. And then when we string those together through condensation reactions, which we will study next, you can take individual nucleotides and make a polynucleotide or a nucleic acid, the polymer. And depending on whether you've built a single-stranded RNA nucleotide or 2 strands of DNA, you could make a DNA double helix.